I'll talk today about, fluorescence based sorting of nuclei for a particular question that we have in our lab. Evan's been a great help in facilitating this approach, and we've been applying it to several projects now. Our lab studies endothelial cells, which are a small relative component of many tissues, but are really key for allowing immune cells to infiltrate into the tissue and deciding when and where those immune cells get into tissues. So we've been interested in them in cardiovascular disease, host versus graft injuries. We've been interested in them in neurodegenerative disease. We've stayed away from cancer biology for now, but I think that there's probably applications of the same type of approach in cancer biology as well. So our lab has been focused on alternative splicing, post-transcriptional regulation of RNA transcripts in endothelial cells specifically. And this is built on some work I did in my postdoc in Richard Hines' group at MIT, where we were looking at the changes in endothelial cells that line blood vessels at regions that are exposed to disturbed flow. So these are branch points. These regions are where plaque develops in arteries. And over time, those plaques break away and cause downstream clots and strokes and heart attacks. So the endothelial cells line those blood vessels and are a key determinant, again, of whether immune cells get into the plaque and how large and inflamed that plaque becomes. So in work that I had done, we looked at splicing patterns in activated endothelial cells. We found a large set of splicing patterns that were responding to innate immune cells when they're recruited to the endothelial cell layer in these points. And we believe that these splicing changes were rewiring the endothelial cells and making them more pro-inflammatory. And so in projects that I've been looking at in my own lab, we've been looking at this type of pro-inflammatory change in state in the endothelial cells in neurodegenerative disease. And a major reason we've been looking at that is that there's a lot of RNA binding proteins in post-transcriptional regulation known to be familial mutations leading to neurodegenerative disease. And most of the focus has been on neurons, which are probably the major component of the brain and actually fairly easy to look at in the brain because they're pretty large cells, whereas endothelial cells are flat sheets and they're typically harder to look at in brain tissue. But based on some of the data that we had acquired, we thought that some of these RNA binding proteins, which were dysregulated in neurodegenerative disease, might be dysregulated in the endothelial cells, which is now being recognized as a critical component of the early breakdown of blood-brain barrier that in many cases precedes some of the later things that happen in neurodegenerative disease, like the accumulation of amyloid fibrils. And so our general hypothesis is that there were some systemic defects in TDP43, for example, but other splice factors as well. And then some of these things were associated with some of the blood-brain barrier defects that occur with age. So in order to be able to study that, we need to look at endothelial cells and look at transcriptional changes in the endothelium in human disease samples. So the NIH Neurobiobank contains thousands of frozen brain samples, and then there's a lot of others that can be made available. Typically, it's very difficult to get fresh brain tissues from different states of neurodegeneration, for example, that are well annotated. But a lot of these samples exist and are stored frozen. So a key question for us is how do we unlock this information from endothelial cells in these stored frozen brain tissues? So one way that this could be done is by looking at endothelial cell enriched transcripts. So for example, in perturbation of this one RNA binding protein, we find a lot of differentially regulated transcripts, 304 of those, which are regulated in the mouse. And then we subset those for those that might be in the whole brain, but really enriched in the endothelial cells. So we can look at the signature changes in those genes in the whole bulk RNA-seq from brain to see if those things are changed. So this is kind of a first pass way to look at that. And we see some good correlations between the things that change in bulk brain tissue and the things that we're changing in our endothelial cells in a mouse model. But obviously, this is not really what we want to do. We're cutting out most of the things we're looking at because they're expressed in multiple cell types. So we really want to look specifically at endothelial cells. And so again, this is a challenge. And this is a challenge because there's only about 2% of the cells in the brain are endothelial cells. And so there had been a set of experiments using single cell technology to look broadly at the cell types in the brain, isolating these cells and doing single cell analysis. And this was a group, Sci Lab at MIT that had done this. And they did this for a large set of samples. And what they typically found was that endothelial cells are about 0.2%. So even less than the 2% you predict if you did immunostaining and counting of cells. And the reason is that they're probably actually being depleted in the process of sorting out these bulk cells for the experiment. Endothelial cells tend to be very fragile. They're found in large vessel structures, which often don't pass through filters, for example, on the way to sorting cells. So a lot of these were lost along the way. So in the end, these experiments, which were pretty comprehensive, only ended up with about 20 endothelial cells per 10x experiment. And since these 10x experiments are really pretty expensive, it's a very, very low yield of data for the cost. And so we wanted a way to get some better and more informative data from the samples. So another way that had been developed, and this was developed actually after we started our own efforts, was to isolate vessel fragments. So you can isolate vessel fragments from frozen brain tissues. And this has a lot of different steps. Basically, you douse and homogenize the brain, isolate large fragments over a filter, wash those fragments, and then mash those fragments through a strainer, eventually isolate the nuclei, and then sort those nuclei. So this has been a very helpful process. It does isolate a lot of cells from vessel fragments, but it is also very time intensive. So for each vessel that you do, it's taking about at least an hour of prep time. And there's a lot of steps where you could lose a large number of cells through the process. So our approach was different. Our approach was to use, we had seen that nu-N was used to isolate neuronal nuclei, and we hypothesized that we might be able to use an endothelial marker to isolate endothelial nuclei in the same way. And so looking at published data banks, we could see that ERG, a transcription factor, is highly expressed and specific to endothelial cells in the brain. The other thing I knew, because of my background in endothelial cell biology, was in the retina, people had often used ERG as a specific marker for nuclei in endothelial structures in the retina, which are going to be very similar to the structures in the brain. And so here you can see very nice labeling with an ERG antibody, for example, in this whole mount of the retinal vasculature. So all these little red dots, those are endothelial cell nuclei. So it's really comprehensive labeling of endothelial cells. And the green label here is EDU labeling, or proliferative cells. So based on these two pieces of information, we thought that in the brain, there's going to be likely very good labeling with the ERG, and then it'll be very specific for endothelial cell nuclei. And so we wondered if ERG would be able to mark these nuclei for isolation by flow cytometry from nuclei isolated from frozen tissues. And so this is now where we really began to get some help from Evan and Lee in our flow core at UConn Medical School. And they've been really critical in allowing us to develop these approaches, helping us to think about the right controls to run and how to separate these nuclei out so we have a very pure population. So some of the first experiments we did were just to take human cell lines, human epithelial cells, 293 cells, or human vein endothelial cells, umbilical vein endothelial cells, or HUVECs, and stain. And again, as expected, we saw a very high proportion of these HUVECs staining with the ERG antibody, whereas we didn't see staining in the epithelial cell line. And we can do a mixture of these and pull out the endothelial cells based on the ERG expression from this mixed population. So this suggested, based on these in vitro experiments, that this might work. And so then we did a quick experiment where we took a sample of brain tissue, frozen brain tissue, and we isolated out, after homogenizing the brain tissue, we isolated out the single nuclei by DAPI signal here. You can see that as the 10% population. And then that was split into nuclei that were stained for nu-N in one channel or ERG in another channel. And so what you'll note here is that there's a good population of neuronal nuclei marked by nu-N, about 34% in this case of the nuclei coming from that brain, which is relatively proportional to the number of neuronal nuclei you'd expect. And nicely, we see about 2% of this ERG high population. And that's, again, about the right percentage you'd expect for endothelial cell nuclei. And you will note that there is an ERG low population here, which I'll get to a little bit later on, which is also interesting and also very relevant to our studies. So at this point, I started to get some help. So Amy had helped me through the beginning of most of this project. Amy is lab manager and technician in my lab. She's been working with me for a long time. And in the summer, Jordan Silva joined. Jordan was an undergrad at Harvard, and he had come to work in the lab over the summer. And over that summer, we did a lot of the following work to establish some proof of principle. And Omar, who's an MD PhD student who joined the lab recently, helped to really complete some of these studies and perform some informatic analysis on some of the samples that we're getting. So our basic protocol, the protocol I just showed you to isolate these nuclei, is pretty quick. We place frozen brain an easy lysis buffer, which will lyse the cell membrane and allow the nuclei to be popped out of those cells. This is a buffer that many people are using to isolate nuclei from tissues. Put in RNase inhibitors to prevent RNase cleavage. And we homogenize in a bullet blender. This is something that other people haven't been doing, but we found that works very well. It's very quick. So this bullet blender puts metal beads into each tube and basically bashes this tube back and forth. The metal beads homogenize the tissue. And this gives a very good homogenate within just a couple of minutes, all done at 4 degrees. And we can spin down the nuclei from that. And they can wash and stain those nuclei and sort based on these different populations I mentioned. So the overall protocol is very, very quick and very easy. We can do this in parallel with many different samples, and with small amounts of tissue, we can get a very high yield of single nuclei. And so I compare that very favorably to the extraction of these vessel fragments, which is a much more time-intensive procedure, although it does have its own benefits. So the questions for us then are, is this effectively enriching endothelial cell nuclei? Are there similar numbers of nuclei isolated between brain samples? Is it compatible with single nuclei workflows? So can we take these nuclei and then do a 10x experiment on the nuclei that we've isolated? Are known endothelial cell transcripts being enriched when we do this? And then finally, can full transcript information be obtained? Remember, our initial questions are to look at RNA splicing. So this is something that really is hard to get at with single cell technology. It's hard to get full length transcripts with single cell technology, but we felt maybe this approach would give us a way to do that. So first, what we did is we took a number of samples from this NIH brain bank and performed the same procedure. And what you can see across these different samples, some are from patients with dementia, some are old, and some are young. We get a very reproducible percentage of the nuclei that were in the ERG high group, in the ERG low group, or in the new N group. So the variance is about 1 to 3% for these different ERG high and ERG low groups, and about 20 to 50% for the new N group. So this suggests to us that we're really efficiently isolating nuclei consistently through these brain tissues. And this was an important thing for us to know, because we were going to apply this technology to look at different disease states. So we think that we can check this off. So the next question then is this compatible with single nuclei workflows? And so this is ongoing work now that we're doing in our lab. The MD PhD student I mentioned earlier, Omar, has been doing a lot of this work with a very talented gap year student, Jordan Taberski, and they've been applying this approach to now look at whether these nuclei can be integrated into single cell workflows. And so the answer is yes, we can. So these are all individual donors, and these are the UMIs per cell that we're getting from these multiple individual donors. So we're getting about 5,000 UMIs. UMIs are unique molecular identifiers indicating individual transcripts, and we're getting about 2,000 to 3,000 genes coming from each of the cells that we're looking at in these experiments, which are very comparable to other types of single nuclei experiments. So we're able to enrich, we're able to mix cells in the proportions we want, so we can take ERG high, ERG low for the reasons I'll mention a little bit later on, and we can take new N negative, for example. And so when we do that, we get the correct mixture coming back out. So this is now 10x UMAP data. So each of these little dots is a cell. And so you can see a cluster, a nice cluster of endothelial cells, different endothelial cell subtypes. We can see some, it's the microglia actually, so I kind of foreshadow what I'm going to show you later on, but the ERG low group is actually turns out to be microglia. And then we have a lot of the other cell types that are coming from the brain that are coming from this new N negative group. So importantly, we can control the population. So we're no longer looking at 0.2% of the 10x data coming from endothelial cells. Now we can force it to be 50%, we can force it to be 70%, or whatever we'd like. Make sure that we're focusing on that cell type and really seeing differences in that gene expression. That allows us to get a lot more out of an individual 10x experiment. We end up putting multiple samples together in 10x experiment, hashtagging them, and getting a lot more data out of each one on endothelial cells. So it is compatible with single cell workflows. We know that, and we're in the process of generating some really exciting data using that. And then our endothelial cell transcripts enriched. So the 10x data would already suggest that they are, but we've done some additional data. This is from bulk sequencing of the ERG high population. And this is whole RNA-seq. And we compared that to a benchmark of whole cell RNA. So basically what you're looking at here is each dot is in a transcript now. And some of these transcripts are endothelial cell specific, or at least highly enriched in endothelial cells. And this is showing you the relative enrichment as log fold change on the axes in either whole cell or the ERG sorted ERG high nuclei. And you can see that the endothelial cell enriched transcripts are highly enriched in both the sorted nuclei and in whole cells in the benchmark sample that we're comparing to. So we think that this procedure is enriching transcripts just as well as whole cell isolation. And we can look at that a different way here. We can look at enrichment relative to the transcripts that are seen in other cell types here. So we can see a very strong enrichment of endothelial cell specific transcripts. Now looking at bulk RNA sequencing from the sorted population. So looking at particular transcripts now, and this really gave us a lot more confidence in this, just looking at a few transcripts, you can see that von Willebrand factor, you can see is highly enriched here in the ERG high population. This is a canonical endothelial cell transcript. Cadherin 5 or V-cadherin is another highly enriched transcript in the ERG high population. And then I mentioned that ERG low population, microglia population. You can see in that population, we have some enrichment of CD45 and I beta alpha or AF1. So these are transcripts now which we'd expect to be found in the microglia population, and that is what we see in that ERG low population. So yes, known endothelial cell transcripts are enriched. And so then the final question is, so in the bulk sequencing, I just showed you that we're highly enriching endothelial cell transcripts. Now, can we obtain full transcript information? Can we look at splicing patterns in those nuclei that are endothelial cell nuclei? And so this data is showing us that we can. Here's reads now mapped against the genome. So each of these little blue lines is showing you there's a read mapping between one exon and another exon. This is a very large extracellular matrix protein that I've worked a lot on historically. And here you can see that there's many reads that are branching in between different exons, which gives us the ability to look at and count the number of reads that are spanning between an exon that might be alternatively included in the transcript and those that are constitutively included in the transcript. And by looking at the relative ratio of the inclusive or exclusive reads that that show you whether that particular exon is being included in the final transcript, you can get an assessment of splicing frequency in that transcript. So we can look at isoforms in this way and look at changes in splicing patterns in this way. Again, the number of genes that we were detecting with splice reads in each sample from this bulk RNA sequence was very high across different groups. And even from 100 nuclei, we can see that there's a large number of genes that we can detect with differentially spliced transcripts or splice reads in them. So we can indeed obtain full transcript information as well using this approach. So in summary, and I'm going to be finishing a little bit early, which is probably good since I'm the last talk of the day. We can use ERG to specifically mark brain endothelial cell nuclei. We can sort this ERG high population to enrich endothelial cell transcripts. The isolation is reproducible across age and disease states that we've looked at: Alzheimer's disease, ALS, and frontotemporal lobe dementia. It allows bulk analysis of the full transcripts, which is important because we can do this splicing analysis that I mentioned we were interested in. And it's compatible with single nuclei analysis. So we can take these sorted endothelial cell nuclei and put them into a 10x workflow and start to look at subsets of the endothelium by really concentrating on endothelial cells coming from the brain tissue. So I should mention that for most of these tissues, our tissue input is about 50 to 100 mgs of tissue. Some of the more exciting stuff is now that we've shown this really works well in brain, we've started to move beyond brain to other tissues, exploring smaller amounts of input material. So we've found that this works, for example, from small biopsies as well. And so I think in other tissues, it's going to have a particular strength because in brain, although you can enrich by isolating these microvessel fragments, that's very, very difficult to do in other tissues that are going to be fibrotic, for example, like in heart or muscle. It's going to be very difficult to isolate large vessel fragments in the same way. We think that the ERG will give a marker in those tissues that will allow isolation of endothelial cell nuclei from those tissues in the same way. So really where there is no alternative right now to isolate endothelial cell nuclei and concentrate those and look at them specifically. So again, we think this is important because endothelial cells are critical for the infiltration of immune cells into different tissues. So we think this is going to be likely to be an important tool that could be used by other people in their own research questions. So most importantly, I want to give acknowledgments to the people that contributed to this work. This particular lab photo is a little outdated, but I wanted to put it here because Jordan Silva is in this picture, and he did a lot of this work over the summer with Amy Kimball and myself to show this method was working. And then Omar, who's not in this picture, but I showed you earlier, has come into this project more recently and is now doing some single cell analysis using these isolated nuclei. And then most importantly, I want to emphasize Evan and Lee in our flow core who have helped me as I've developed these approaches. I hadn't sorted nuclei before, and they've made it easy and efficient for us to do this and have been putting up with us. Again, I think this is a common theme probably with flow cores, people running late with samples, people not having everything ready in time to get their stuff on, or not thinking all the way through about the different controls they needed to run. And so Evan and Lee have been really crucial to the success of this project, which has resulted in grant funds from NINDS and other sources. So with that, I'll end and ending early again. Hopefully there's plenty of time for questions then, and I'll let you get on to the next phase in the conference.
Thank you very much, Patrick. Can you hear us well?
Yeah. I can hear you. Yep.
Can you hear us well?
Yes, I can.
Okay, perfect. Wonderful talk. Okay. Can you still hear us?
Yes.
Okay, perfect. Sorry about that. Do we have any questions in the house? Okay, do we have any Zoom questions? No? Okay, thank you so much, Patrick. Okay.
Sure.
One.
Hi. Just since you already have the neurons labeled with new N, have you thought about comparing gene expression in the endothelial and neuronal cells in different diseases?
Yeah, I think when we're doing the mixing now, what we're currently doing is putting a small amount of neuronal nuclei, not 30% obviously, but a small fraction of neuronal nuclei in. And what we're most interested in is the interaction between the microglia and the endothelial cells. So those are the main cell types that seem to be driving the inflammatory phase of neurodegenerative disease. And so we're mainly focusing on the interactions between those two cell types, but we can look at, for example, molecules that are expressed by one and have receptors on the other. And so we can look at crosstalk between those two cell types. So yeah, that's a good question. I think that's one of the advantages of the single cell data is you can do that type of analysis. So anyway, so I think that from one set of single cell data, there's quite a lot of downstream analysis to do, and it'll keep us busy for the next few years, I'm sure.
Just an easy technical question. The bullet blender, is that a one-time use with the beads and such? Is there an expense with that? And just kind of how does that work?
Yeah. So the instrument itself, I think, was relatively cheap. I think it was under 5,000. And then, so the instrument is basically a bunch of tube holders, and it's got a bar inside, and the sample goes in with the beads inside. And the beads are one-time use. I mean, I suppose you could extract the beads and wash them and clean them and put them back in, but we haven't. We've just tried to avoid any overlap between samples. So typically you'll buy like a container of beads, which is also relatively cheap. I think maybe $10, something like that. And you put a few beads into each tube, and the tube then goes into the machine. The bar swings around and hits the tube and sends these beads flying all over inside the tube, disrupting the tissue. I think the advantage for us is that when we're working with brain samples and things like that, everything is contained within these tubes. So we can open them up outside in the containment hood, for example. Each tube, you can put multiple tubes into the machine at once. I think we've done as much as 8 in the machine at once. And you'll homogenize those. They can all be done together at 4 degrees, pull those out. And if you have another batch, you can stick that into the machine. So the machine, you buy once and that's all you need to do. That's your capital equipment. And the only other stuff that you have to buy after that are the tubes and the beads.
Thank you very much.